Hello and welcome to this video podcast for Biology 211 Lab. In this podcast, what we will do is we'll look at wild-type worms, that is worms that don't have a mutation in the gene we're interested in, and we'll also look at worms that have a mutation in a gene that we're interested in. And then we'll see how each of these worms perform in a chemotaxis assay. Quick overview of Lab 2.2. Now there will be questions from this week's quiz that come from this Lab 2.2. We're going to go over the lab report 2 that you're responsible for this module. We'll talk a little bit about the format and expectations. We'll talk about null versus alternative hypotheses and some other things as well. I'll give you a brief background on chemotaxis, what it means, and how we're going to study it in the worms. And then also a little bit about motor proteins. If you remember from cell biology, motor proteins are important for the cytoskeleton, and dyneins, they are one of those motor proteins. There will also be an opportunity for you to design your own experiment, and then the experiment you design, you'll analyze results from similar experiments next week. And then you'll analyze your data from the chemotaxis assay, and then make a figure, which will be a part of your lab report that you need to write for this module. What's due from this lab? Worksheet 2.2, parts 1 through 3 are due for this lab, and due on Thursday like most of our other assignments are due. A little bit of framework of where we've been and where we're going. In the last lab, we talked about the BARGMANN paper. We introduced C. elegans. We talked about wild type and mutant forms. Today, as I said, we're going to talk about the chemotaxis assay, design your own experiment, and analyze the data from a previous experiment. And you'll work on figure 1 and methods for your full lab report. And then next week you'll see the first day here, you'll analyze data for a design your own experiment and design figure 2. And then on the next 2 days, these are days mainly meant for writing. You have the full lab report. We want to make sure you have plenty of time to fit into your schedule to write the full lab report. The peer editing here and here, I'm going to make optional for the lab report. With this being an online class, I can see how it was difficult to get 2 people to peer edit your introductions for module 1. However, I really think it's important that you do the peer editing, and so I still encourage you to have somebody look it over. Like I said before, everything I've ever written that's been published, or even the things that haven't been published, I always have 1 or 2 people peer edit what I've written. It makes for a better paper. In your case, it makes for a better lab report and a better grade. So make sure you have somebody peer edit your work. All right, so plenty of time next week to get the writing done for this project. Okay, a quick overview of lab report 2. This lab report will include an introduction, a methods, a results, 2 figures with legends, a discussion, and references. Between Biology 111, 112, and 211, you've already written all of these parts, but now we're just going to put them all together into a single lab report. Review pages 75 to 80 in the lab manual for descriptions of what to include and how to format. It's all laid out there fairly nicely. In fact, why don't we just go there real quick? Okay, we're on page 75 down here, and it'll lay out everything we're going to do, just like I said previously. It tells you all the parts of it, highlights of what each section is. Your writing manual will contain a little bit more details, and actually pretty good details. You'll see a rather long and detailed rubric. It's detailed because we want to provide you with all the information you need so you can get the best possible grade on this lab report. So make sure you look at this rubric, have somebody peer edit your paper, and go through each of these steps and circle where you're strong and where you're weak on your paper. We give you plenty of time next week to do that. We're not requiring the peer editing, but it's still very important to do. Okay, let's go back to the PowerPoint. As I said just a moment ago, and at the beginning of this video, we are writing the report in sections and incorporating peer editing to help improve everyone's writing and grade. So again, I'm sounding like a broken record here, but really take advantage of this time you have and the peer editing to make a very strong paper. While writing your report, it will include data from Lab 2.2, that's today, and Lab 2.3, which we'll do next week. You're going to start working on lab report today by drafting a brief outline using the rubric that I just showed you that's in your manual. You don't have to turn it in, but just because we're not asking you to turn it in doesn't mean you shouldn't do it. So please do it. This will make writing the full lab report much easier for you. Collect and analyze data, and we'll talk about that in a moment. You'll also prepare figure 1 and its legend, and quite honestly, I'd go ahead and get that written this week so you don't have to worry about it next week. And then you'll start working on the methods section, and you should be able to get a good part of the methods section done. Alright, so to write the method section, just to remind you, you've written one of these before in a previous class, but go back to your Macmillan writing manual and look at pages 75 to 81. Now those page numbers may be a little different depending upon the edition of the manual you have, but if it's not exactly page 75 and 81, it will be close. And so you may have to just kind of skim a few pages before and a few pages after to find information on the methods. We ask that you write the method section in the passive voice and in past tense. This is how methods are written because you're describing things that have already happened. For instance, you may write, we performed the experiments by placing the worms in the center of the plate. Write in a paragraph form, that is, don't make a bullet point list. When we do the poster for the yeast experiments, you'll see that we prefer some bullet points. But in a lab report or a paper to be published, we write the methods in a paragraph form and not a bulleted list. Use the personal pronouns I or we, if you're talking about yourself and your lab partner, rather than the investigator or the author. It used to be, and maybe in some science classes you're still taught this, but we really embrace using the personal pronoun in many parts of biology. Within the method section, include at least two subheadings. The first subheading will describe the experiments from 2.2, and the second subheading will describe the experiments from 2.3. Do not call them Lab 2.1 or Lab 2.3. Rather, they should be descriptive summary sentences explaining labs 2.2 and 2.3. That is what you did in those labs. While I say there should only be two, you may come up with a third or a fourth, and that's okay. While I said there should only be two subheadings, you may come up with a third or fourth, and that's okay. Within each of these subheadings, you should include laboratory procedures and description of the statistics used. Make sure, as I have already shown you, you go back and look at the expectations from the grading rubric on pages 77 through 80 in your lab manual. So for instance, I'm only showing you the 100% column because that's what we want you to shoot for here, of course. The methods content should have all of these pieces of information. Make sure that your methods don't contain extraneous information, and you omit those. And the rubric gives you some nice examples of things to not include in your report. The tubes were labeled. Gloves were worn. The machine was allowed to warm up. These are all assumed. We assume that standard laboratory procedures were followed. I always think of a paper as something that costs money per every word you use. And so you want to use as few of words as you possibly can without losing the impact of the statement. Methods writing, subdivided into sections with clear and appropriate subheadings. So if you put Lab 2.2 and Lab 2.3, you're going to lose points on that. Write it in the right tense. If you start writing it in the present tense or the future tense, you will lose points. All right, let's talk a little bit about the null and the alternative hypotheses. These are two terms that get thrown around quite a bit, and sometimes they're used correctly and sometimes they're not. When we say the null hypothesis, that says that there's no statistical significance between the two variables in the hypothesis. And we'll go over some examples in a moment. It's called the null because researchers work to nullify this hypothesis. With the null hypothesis, the experimenter expects to see no difference or effect. The alternative hypothesis, or the research hypothesis, is the opposite of the null hypothesis. With the alternative hypothesis, the experimenter expects to observe some differences or effect. Gives an alternative to the null in what the researcher attempts to prove. So in a properly laid out experiment with a hypothesis, the null hypothesis is stated and we try to essentially disprove it. And by disproving it, we give strength to the alternative hypothesis. And we will eventually come around to supporting the alternative hypothesis. So let's look at some examples. People used to believe the world was flat. Scientists had to disprove the null hypothesis to accept the alternate hypothesis. So the null hypothesis is the world is flat, because that is what people used to believe. And I've been told there are still people who believe that. But at one time, most people believed that the world was flat. So the alternative hypothesis is that the world is round. So what you do is that your experiments are set up to test this hypothesis. And if you can't find support for this hypothesis in your experiments, then that brings support to the alternative hypothesis. Another example, a new teaching method is developed that is believed to be better than the current method. The null hypothesis would be the new teaching method is no better than the old one. If after attempting to support this hypothesis. You can't. The experiments do not support this hypothesis. That leads the researcher to have more support for the alternative hypothesis. That is, the new teaching method is better. Let's do a practice. If you do an experiment to see if a medicine works, consider these two hypotheses. A, the medicine makes people healthier. The medicine does not work. Go ahead and hit pause and think about which one of these is the better null hypothesis and which one is the better alternative hypothesis. And then hit play, and then we'll answer this question. Welcome back. What we see is that B, the medicine does not work. That's the null hypothesis. That is assuming that the medicine does not work. The alternative hypothesis is that the medicine makes people healthier. So they essentially nullified this experiment in this example here, which led support to the alternative hypothesis. If you do an experiment to see if people like chocolate or vanilla ice cream, what would be the null hypothesis? And then what would be the alternative hypothesis? Go ahead and hit pause and write those two out. Okay, welcome back. So a good null hypothesis with this would be that people like chocolate and vanilla equally well. And a good alternative hypothesis would be people like chocolate ice cream more than vanilla, if that's what the data eventually supported. That would be the correct data in our household as well. Okay, let me give you some background on chemotaxis and motor proteins, particularly dyneins. Chemotaxis is a general term used in biology to describe the attractance or sometimes the avoidance of particular chemical substances. Again, chemo for chemical, taxis for movement. So what cells in worms might be involved in controlling attraction or avoidance of chemical substances in their environment? You may have thought neurons, and that would be correct. Neurons are the cells that are going to be the control center of what the worms do in response to certain chemicals. Now, of course, there are other cells involved. Muscle cells are involved because the worm has to move, and other cells as well. But neurons are ultimately responsible for the worm to know which way to move. What proteins are needed for signaling? Well, ultimately, that signal has to be moved along the neurons in some way or another. And so we know microtubule proteins are going to be important, particularly motor proteins. And if you remember from Biology 112, kinesins and dyneins are microtubule motor proteins. Kinesins move from the cell body to the axon terminal, and dyneins are motor proteins that move that cargo from the axon terminal to the cell body. We're going to focus on dyneins today because the mutant that we will be studying is a dynein gene. So here's just a diagram of what I just explained, showing kinesins moving from the cell body to the axon terminal, and then dyneins taking cargo back in the opposite direction from the axon terminal to the cell body. As I said, the mutant we will work with today is a dynein mutant, and that dynein mutant in worms is called CHE3, CHE-3. In this mutant, dynein proteins will not function. And so we would predict that they will not be able to do chemotax. All right, now, in the last lab, you read the BARGMANN paper and analyzed that. And from that paper, we were able to determine that wild-type worms are attracted to isoamyl alcohol. So we have that information to use when we design this experiment. So one might predict then that dynein and kinesin mutants are defective in chemotaxis. The paper included CHE3 and other mutants. So we're going to look at data using wild-type and CHE3 mutants before we design our experiment. So in today's experiment, you're going to look at data comparing wild-type and CHE3 mutant worms, and then we'll show you data with CHE3 mutant strains, and these data will confirm that CHE3 strain is defective in chemotaxis. So we can think of the positive control as the wild-type worms with isoamyl alcohol. We would expect these worms to chemotax towards this favorable condition of the isoamyl alcohol. The paper showed about an 80% chemotaxis index. We would expect our chemotaxis index to be in that range as well. It may not be exact, but it should be in that range. The experimental condition, that was the CHE3 worms with isoamyl alcohol. We would expect that chemotaxis index to be substantially less than 80%. I'll provide you with the class data so that you can use that to complete Figure 1 in Lab Report 2. Now, you're not going to be doing the actual experiments in the online lab, but it's important to note some safety features. Sodium azide, which is a chemical that we use that is a poison. We put that in a part of the plate, which I'll explain in a moment, that will cause the worms to die. Likewise, it is also not very good for us to consume this because we don't want our cells to die, and we don't want to die. So when students handle this, or anybody handles this, they always wear gloves. As is true with a lot of sciences that use plates, if it's a microbiology experiment or worms experiment in this case, always be careful not to puncture the auger because that can cause certain kinds of growth problems. And in this case, the worms may find that hole and just kind of burrow into that hole. And that kind of ruins the experiment if all your worms are burrowing into holes and not moving to or away from the chemical you're studying. Okay, so let's go ahead and talk about how one would set up a plate for this experiment. And this is important for you to know as you write the methods section. We take a petri dish, just like you might any petri dish in a microbiology lab, but we're working with worms, and they use the same kind of dishes. And you want to mark the plate as shown here. This is important so that once you begin to study the results, you know where each section of the plate is. In the middle, we're going to mark this the W spot. This is where we'll place the worms. Up here, we'll mark it with an X. This is where you'll place the attractant. And down here, we make a dot, a large dot, and that's where we put the carrier. Let's spend just a moment talking about what I mean by carrier. The carrier is the solvent used in the attractant. So in this case, the attractant is isoamyl alcohol, but the solvent used is ethanol. So we'll put ethanol down here and isoamyl alcohol up here. We want to do this because we want to make sure that the reason the worms move towards the isoamyl alcohol, if that is what they do, is because of the isoamyl alcohol and not because of the solvent that is in the mixture, which would be ethanol. So this control is really important. In addition, as you see here, the word azide for sodium azide is added here. On the previous slide, I explained to you how this is a poison and it will kill cells. So when we add it here, we know that when worms get here, they'll die. The reason that's important is because if we didn't have it, there's no way we would know at the end of the experiment, which is about an hour, if worms came up here and then decided to cruise back down here, or if worms started here and moved to the carrier area and then came back up here. We want them to stay in this area, and so the best way to do that is to put this poison in here, sodium azide, and when the worms get there, they become paralyzed. They become paralyzed and they die. So to start this experiment, you add the sodium azide to A and C first, and then you'll add 10 to 15 adult worms to the W spot in the middle. Then you'll add isoamyl alcohol to the A spot and ethanol to the C spot. You kind of want to make sure the worms are moving before you start the timer for the hour incubation. Because if they're not moving, then your experiment is not going to work. And sometimes they get stuck in this big bubble of water and you kind of have to break up that bubble so they can start to move. So you want to make sure they can move before you do the experiment, as opposed to coming back an hour later and they're all in the middle, still unable to move. Then you just wasted an hour. And we don't want to waste an hour. Nobody's got time for that. Incubate 1 hour with the microscope light off. Sometimes we forget and we now have the microscope light on, and now we've heated the plate, so we're testing a second variable as well. And there's no way to know if the worms are moving in one area or the other area or not moving because the plate is hotter than it would normally be. Then what is done afterwards is the chemotaxis index is calculated. We're going to just give you the chemotaxis index, but you should know how to calculate this. Maybe I will even put a question like this on the exam. If I do, I will let you know, so you know to make sure you can memorize this equation. But the chemotaxis index is the number of worms at the attractant pole, that is where we put the isoamyl alcohol, subtract from that the number of worms at the carrier pole, where we had the ethanol, and then divide that by the total number of adults on the plate. So somewhere between 10 and 15. And then you multiply that by 100%, and that is your chemotaxis index. Now I say multiply by 100%, but often the number is also reported as just a decimal. And you'll see that's what's reported on the next slide. All right, so now you'll notice here I'm saying we're moving to part 3, we're skipping part 2. And I want to talk about part 3 now because that's the actual analysis of the data. If this were a face-to-face lab, there would be an hour incubation. And during that hour incubation, we move to part 2 so that we can be more efficient with our time. But since this is an online demonstration of the lab, we can just move straight to part 3 and analyze the data while we're thinking about it. So these are our chemotaxis indices shown here, and these are the mutant chemotaxis indices. Record these data for both wild type and key 3 in the table found on your worksheet 2.2 part 3. You'll want to write a null hypothesis. Recall that a null hypothesis says that there is no statistical difference between the two variables in the hypothesis. This means that the experimental key 3 will not be different than the control. That is what the null hypothesis would say. So we could write the null hypothesis as this. Key 3 chemotaxis index will not differ from the wild type control. Now, of course, these would always be written before you do the experiment. Now, of course, unlike shown here on this slide, these hypotheses would be written before this experiment is actually done. And then remember that this worksheet 2.2 is due Thursday at 11:59 p.m. Alright, so now let's think about how to analyze these data. I would recommend using Excel to analyze the class data and to answer the questions on the worksheet. The reason I recommend using Excel is because if you look in your lab manual, pages 69 to 72, we list step by step what you need to do in Excel to generate these analyses. So you're going to calculate a t-test. Look on page 70 for how to do that with Excel. Read how to interpret the p-values linked to the t-tests on page 71. Create a bar graph with error bars included. How to do that? Look on pages 71 through 72. Have a TA initial your worksheet. In the online version of the lab, this isn't as practical to do. So we're going to trust that you just do this so that you're not next Thursday evening scrambling to get this done at the last minute. I'd recommend that you check the lab rubric to make sure you understand how this is going to be graded, so when you make it, it's made correctly the first time. You'll have two figures, one from this experiment 2.2 and one from the design your own experiment that we'll do next week. It turns out, because you're all working with the same data, that each student's graphs are going to be very similar in this case, and that's okay. We expect that. Work on the legend for the bar graph. Again, this will be figure 1 in your lab report. Each student writes their own legend, so they should be different, but it's okay if they sound similar because it's the same data and it's the same graph, essentially. And then I want you to begin working on a draft of your methods for lab report 2. Each student writes their own method section. Each one should sound different and be independent, but it's also common that there will be some overlap in these methods because method sections just tend to be that way. So from your rubric, I'd like you to read those expectations so that when you begin writing these different parts, you understand what they should include. So let's look over here. In the example that we talked earlier about chocolate and vanilla ice cream, so we have these 3 examples of a title for this figure. Go ahead and look these over and select one of them. Hit pause if you'd like. The best title for this would be People Prefer Chocolate Ice Cream Over Vanilla. Because remember, as we talked about figure legends during the plant lab, the title should describe the take-home point from that figure. And as you look at the figure, more people, wisely I should say, chose chocolate ice cream over vanilla ice cream. And so that's what the title should tell you. Now look at this figure with its legend, and I want you to come up with 3 reasons that this is wrong. You might come up with more, but at least 3 reasons that are wrong. Hit pause and then hit play when you're ready to see the answers. Welcome back. The first one is that the title does not summarize the take-home message. Again, they did not use the suggested title that we have up here. Chocolate versus vanilla ice cream is a true statement. That's what this figure is showing, but it's not telling you the take-home point. The actual results are stated in the legend. What should be in the legend are details about the experiment. The results themselves are shown in the figure, so you don't need to repeat them in the legend. They have figure 1 plus the title highlighted. Only figure 1 should be highlighted. And so this is a better way to write that figure legend. The reason we show this as an example is because these are common errors made by students when writing their full lab report. Now let's move to part 2, the part that if it were a face-to-face lab, you would have been doing during the incubation. But now let's look at this. This is design your own experiment. Now unfortunately you won't be able to run the experiment, but we want you to have the experience of designing the experiment, and then we'll provide you with data afterward. Clearly this is not the way science is usually done. Data are gathered from the experiment, but we want you to be able to analyze the data. So read lab manual 2.2 part 2 and complete the worksheet 2.2 parts 1 and 2. In your team, develop a research hypothesis regarding chemotaxis to test based on published literature. You can use the BARGMANN paper or some other paper. Note that the research hypothesis is like the alternate hypothesis. This is not your null hypothesis. Design an experiment. Using the guidelines in the lab manual and worksheet 2.2, each team gets 2 attractant plates, and you can only test one variable. Think about how you would like to design the experiment using these 2 attractant plates. And then make sure you complete worksheet 2.2, parts 1 and 2. Remember, this is due Thursday at 11:59 p.m. Possible reagents for you to consider in designing your experiment. You could think about a temperature variable, 23 to 31 degrees, or maybe a lower temperature of 15 degrees. Don't go lower than 15 degrees or over 31 degrees, because these are temperatures that C. elegans will not move in. Possible attractance plates. You could have regular attractance plates, similar to the experiments described today. We could put food in the middle where we place the worms to see if they prefer to stay in with the food or move towards an attractant. You could also put food in one of the attractant spots. These are just some examples. Possible worms, wild type, or key 3. Now with the face-to-face lab, we only give these 2 options because these are the worms we have readily available for the experiments. However, this being an online lab, what we could do is allow you to pick a different worm by looking in the literature, and you may find it'd be fun to look at this mutant or this other mutant. That would be fine. Just make sure you let us know on your write-up paper. Some possible chemicals you could use. You could use acetone. It's known as an attractant to wild-type worms. Isoamyl alcohol. Now, you wouldn't want to use it just as you used it today, but you might want to use different concentrations of it if you wanted to use isoamyl alcohol. Sodium chloride, sodium azide. You could look at other alcohols or other kinds of chemicals. And these are just some examples. If you have questions about this, make sure you chat with your TA about if it's an appropriate experimental design. Okay, so for next week, Lab 2.3, make sure you read in your manual about Lab 2.3. There will be a quiz about Lab 2.3 next week as well. Read the following sections in your writing manual: results and discussions, improving paragraph, punctuation, and verb tense. Always a fun topic. Lab 2.3, we will provide you with sample data depending on your experimental design. Peer editing of your full lab report. Again, I said earlier this is optional, and it is optional, but it's strongly encouraged. Completing your full lab report. This, unlike the worksheets, will be due Friday. We'll give you an extra day to get the full lab report done at 11:59 p.m. on Friday. Okay, that's all I have for this video podcast for Biology 211 Lab. If you have any questions at all, make sure you contact me or your TA, and we'll address any questions or concerns you might have. Good luck, and we'll see you in lecture lab later on. Bye.